Good morning, and thank you, Dr. Friedman, for the invitation to present some of our work based on surface plasmon resonance imaging that we developed for the detection of living bacteria. Most of the results presented today have been achieved in our group based in Grenoble, in France, where we acquired some experience on the SPRi biochip engineering and processing for several applications such as food safety, biomedical analysis, or environmental controls. First of all, let me very shortly introduce the physical principles of surface plasmon resonance imaging. For several years now, we are using SPRi lab systems developed by HORIBA Scientific. These instruments contain a monochromatic and polarized light source and a CCD camera. They require SPRi biochips made of an high-index glass and covered by a 50 nanometer thick gold layer. This metal is chemically stable in most biological conditions and can easily be functionalized. SPR, to make it short, is an optical technique enabling the follow-up of any optical index change occurring in a 100 nanometer thick layer on the biochip. Indeed, if we change the incident light angle, we may observe a drop of the reflected light intensity when the light hits the gold layer at the resonance angle. The effect is described in the plot in the upper right corner of the slide. This phenomenon is dependent on several parameters like the temperature, the wavelength of the incident light, and the optical index of the medium in contact with the gold layer. Then, if any compound comes close to the surface, it will locally induce an optical index change that can be followed and plotted in real time. As can be seen on the photo in the upper left corner, the biochip surface is about 2 square centimeters. It means we can easily array up to several dozens of different probes by making small features of a few hundreds of microns in diameter. Such multiplexed biochip enables a labor-free and real-time follow-up of a single sample interacting with a large series of probes. In the case of bacteria detection, immuno-based arrays made of biochips functionalized with several antibodies targeting bacterial surface antigens might be used for SPR imaging. As chemically shown here, whole bacteria may interact with one or several antibodies and then produce a specific signal recorded by the SPR imager. Of course, such detection depends on the antibody efficiency as well as the antigen presence and abundance on the bacterial surface. Anyway, if you look at the literature, you may find several publications describing the detection of whole bacteria by surface plasmon resonance. Here are a few examples of such papers. As you can see, several highly relevant pathogens might be detected, such as Listeria monocytogenes, E. coli 157H7, Salmonella enterica, Yersinia, and so on. Another interesting thing about these publications is the limits of detection given in these papers. Indeed, most of the limit concentrations required for detection are in the range of 10 to the 2 up 10 to the 9 bacteria per milliliter. In the domain of microbiological studies, it is very important to keep in mind the real pathogen concentrations that can be found in a sample, whatever it may be. For instance, these levels might be so low that in the case of European food safety regulations, all certified methods must enable the detection of one bacterium present in 25 grams of food. In the case of biomedical analysis, and for instance in the case of sepsis, only few bacteria might be present per blood milliliter, meaning only few pathogens are diluted in several billions of blood cells. In other words, it means the SPR-based methods shown on the previous slide cannot be used if the sample is not enriched in bacteria. Indeed, all certified methods used so far require a pre-enrichment step, meaning a phase enabling the bacteria multiplication in the sample diluted in culture media. The purpose is to increase the bacteria concentration above the admitted limit of detection of the method. This two-step process is done for both food safety controls and blood testing. Usually, the enrichment step lasts 12 to 18 hours and is followed by the analysis step lasting less than 1 hour in the case of PCR, for instance, and up to several days for cultural methods as shown here in the slide. A few years ago, we developed a method enabling to carry the enrichment step on the biochip itself. Samples are processed according standard protocols validated for the enrichment step, which is carried directly in the SPR imager. As the concentration increases and exceeds the limit of detection, the SPR response arises by comparison to control signals. Such one-step method that we named culture capture measure has several advantages. First, it gives an answer during the enrichment step, which significantly decreases the time to response delay. Second, it significantly reduces the sample handling step number and thus decreases the exposure risk for the technicians. Third, a single multiplexed biochip might be used for the detection of several pathogens, which eventually significantly decreases the analysis cost. On this slide, I present some SPR sensorgrams we got when processing several samples spiked with Salmonella enterochides according to the culture capture measure method. Similarly to qPCR, depending on the bacterial concentration present at the very beginning of the experiment, one may measure a characteristic time labeled CT on the plots shown here. This characteristic time will depend on the initial bacterial load, as more concentrated samples will give shorter CTs, while less concentrated samples will require more time to produce a positive response. In the case shown here, 2 Salmonella CFU per milliliter have been successfully detected in less than 10 hours. These data have been obtained on biochips arrayed with antibodies specific to Salmonella enterochides or specific to Salmonella typhimurium, or recognizing both bacteria. The array also contains an antibody specific to a eukaryotic antigen that is used as a negative control antibody. In collaboration with Patrick Fax's group, located close to Paris in France, we developed a series of SPR biochips specific to Shiga toxin-producing E. coli cells, also named STAC. These pathogens all belong to the E. coli species, but are distinguished by their O antigens. Due to their dangerousness, they require biosafety level 3 facilities. In this work, published a few years ago, we showed how each serotype of the most frequent STAC pathogens could easily be detected by SPR at both low concentrations, meaning few CFU per milliliter, or high concentrations, meaning few thousands of CFUs per milliliter. This data proved the method is suitable to search deeper than bacterial genus or species, as molecular antigens expressed on the bacterial surface can be screened by SPRi. In the case of food safety, a frequent challenge is a search for a pathogen that might be present in a sample containing a natural flora. Sometimes the problem looks like searching for a needle in a haystack. As shown in this slide, we adapted the enrichment conditions of the culture capture measure method to successfully detect low levels of Salmonella, about 10 bacteria, spiked in sheep raw milk used for the production of French blue cheese and naturally containing 10 to the 6 up to 10 to the 8 lactic bacteria. This data confirm the SPR-based detection of a specific pathogen may be successfully completed even though the sample contains high concentrations of natural flora. I guess you understood that the detection of bacteria on SPR biochips strongly depends on the antibody efficiency to specifically bind surface antigens. In the framework of a collaboration with the Prestudia company in Paris, in France, several rabbits have been immunized with different Listeria strains to produce polyclonal antibodies, hopefully specific to the molecular serotypes characterizing the inoculated Listeria. The antibody names, Listeria strains we used for immunization, and their respective serotypes are listed in the table in the lower left corner. The antibodies were then arrayed on an SPR biochip, and several Listeria strains of each serotype were individually tested for detection. 184 SPR experiments have thus been carried out with Listeria samples, along with 34 control experiments carried with other bacteria processed on similar biochips. Detection efficiencies are presented in the large table displayed on this slide. These polyclonal antibodies showed sometimes pretty high efficiencies even against serotypes absent from the immunization cocktail. For instance, AB6 has been produced by immunization with 2A, 2B, 2B and 4B serotypes, but also recognized 100% of the 2C Listeria serotype, even though the serotype was not introduced into the cocktail. On the contrary, in the case of AB1, only 27% of the 2A serotype samples are recognized even though these antigens were used for immunization. Actually, the AB1 turns out to be more efficient for the detection of 2B or 4B serotypes, although they were not present in the immunization cocktail. As a conclusion, I can say that the production of polyclonal antibodies may be considered as a quite straightforward way to get access to efficient SPR ligands. But the main limitation here is due to the lack of control in the polyclonal specificity when immunizing animals with whole bacteria rather than with pure and isolated molecular antigens. This data confirms that in such conditions, the polyclonal antibody production may lead to some serocourse reactivity. As an intermediate conclusion, we can say that the culture capture measure method carried on antibody SPR biochips was successfully validated in conditions similar to food industry safety controls. It was tested on fresh lettuce, apples, powder infant formula, fresh and raw milk, ground meat, etc. Several pathogens, either gram-positive or gram-negative, were efficiently detected: Salmonella, Listeria monocytogenes, Enterobacter sakazaki, E. coli O157H7, and several stacks. Actually, we managed to detect all the bacteria we tested so far. The only troubles we met were with Staphylococcus aureus samples. Indeed, as this strain naturally expresses A proteins on its surface, the presence of this pathogen gave positive signals on all antibodies arrayed on the SPRi biochip, even on the controls. It confirmed that in the case of immunoassays, the design, choice, and characterization of the ligand is a critical issue. The complete characterization of each antibody is mandatory to ensure the lowest level of false negative responses. For this reason, we decided to explore new ligands, enabling the SPR-based detection of a wide spectrum of bacteria. In other words, the question we asked was, what kind of ligand could we use to detect the largest panel of pathogens? The main application targeted here is a biomedical analysis of blood or other body fluids expected to be sterile in normal conditions. Quite recently, we started working with antimicrobial peptides as potential ligands. Antimicrobial peptides, named AMP, are natural short peptides, usually less than 30 amino acids long, active against bacteria, gram-positive, gram-negative, or both, but also on fungi or viruses. They are found in plants, insects, and mammals. They are usually highly positively charged and tend to form secondary structures made of alpha helices. More than 3,000 AMPs have been described so far and are listed in a couple of databases. You will find here a link to one of them. Since their discovery almost a century ago and the identification of their role in the innate immune response, many efforts have been made to exploit these peptides as antibiotics. For instance, if you run a search in PubMed with antimicrobial peptides and antibiotic as keywords, you will find more than 2,700 papers published on this topic in 2018. A general action mode admitted for most AMP is an electrostatic and hydrophobic interaction with the bacterial membrane, and their self-assembling into barrels to form pores into this membrane. Interestingly, only few data describe their use as immobilized ligands for bacterial detection purposes. Here is the first set of peptides we rate on SPR biochips. The AMP sequences have been ordered from a local peptide supplier and were quite affordable. The choice of these sequences has been made on the basis of few examples describing AMPs as biosensor ligands. Two peptide orientations have been tested by grafting model sequences either on the C or N end. Well, after AMP arraying on biochips, several bacterial suspensions containing pathogens, similar to the ones assayed on antibody biochip, were processed according the culture capture measure method. As can be seen here on the SPR images of the whole peptide array, all strains got successfully detected, including Staphylococcus aureus. We also observed that the SPR response varied depending on the AMP sequence and on the bacterial strain present in the sample. Such results are even more visible on the next slide, where we plotted SPR sensorgrams for each peptide. These plots thus confirm that the same set of peptides, here I'm talking about only 5 different AMPs and a control, enabled the detection of 5 different bacterial strains, either gram-positive or negative. We got pretty excited by this data, confirming that our approach, based on the use of AMPs as ligands, was suitable for wide-spectrum detection. If we look closer to the SPRi data, we observe that the AMP's interaction profiles are different depending on the bacteria. At this point, we decided to go deeper into the data analysis to test if this peptide set would enable bacteria identification, which is quite different from a simple bacteria detection. This hypothesis was confirmed by multivariate analysis. To do so, principal component analysis was done on SPR reflectivity shift and sensitivity values at the maximum of the first derivative for each peptide. This statistical treatment aims at objectively assess the discrimination values of our method. Each bacterial strain was processed once on an AMP microarray, giving 64 sets of values from all possible combinations of peptide duplicates responses at the surface. These experiments on different bacterial strains interacting with the same set of AMPs gave clustered results as shown on the PCA2D score plot. Those clusters of data points were well segregated from each other according to the monitored pathogen. It thus confirmed the suitability of this approach for bacteria identification, at least for the set of bacteria tested here. Well, as a conclusion of this webinar, hopefully I convinced you that SPR imaging of multiplexed biochips might be used for detection of bacteria. In the case of live bacteria suspended in culture media enabling their multiplication, very low levels of contaminating pathogens are detected. The use of antibody biochips enables a strain and even serotype identification. In this case, the success is highly depending on the antibody efficiency. AMPs might also be considered as interesting ligands alternative to antibodies, as they have several advantages. First, they are pretty affordable and easy to modify and graft on SPR biochips by including a terminal cysteine, for instance. Second, the same set of peptide might be used for the detection of different pathogens. And third, multivariate analysis may give more information such as bacterial identification. We are now working on the method improvement and extension to complex samples such as blood, where peptide stability upon time might be an issue. To finish, let me thank all the people involved in this work, either in our group, in the SimLab, or through the collaborations we had so far. And thank you all for your attention.
In this webinar session, we will talk about the application of Raman microspectroscopy to clinical microbiology. The intent is not to provide an exhaustive review of what has been done in the field. You can find those kind of reviews in the literature. The intent is more to illustrate what can be done in the context of clinical microbiology. This is the outline of my presentation. I will do a very short introduction to the context of clinical microbiology that we have considered, then explain why Raman has been used, and more specifically explain the global system approach that we have used that integrates Raman, but not only Raman, other optical methods and sample preparation. Then provide information about the single microbial cell identification that we have obtained, and the same for the phenotype determination of antibiotic resistance or susceptibility, and show that this has been done not only at the single cell level, but at the level of populations of single cell, which is much more interesting than short conclusions. So first, an introduction to the clinical microbiology context. So we are at the level of clinical microbiology labs. When someone is suspected from having an infection, a sample is taken, so blood, urine, anything. And what happens in clinical microbiology labs, less classically, is that this sample is treated onto a solid growth medium. And the whole is incubated until each bacterium of the initial sample has grown to a colony, so that's 10 to the 8th, 10 to the 9th clonal individuals from the initial bacterium. And then one or several of those colonies are chosen for subsequent identification or susceptibility testing. For identification, now the very classical method is Malditoff. So a colony is streaked onto this kind of plates. The wall is inserted in this Malditoff system, and identification is known within a few minutes now. So the whole process is about 1-2 hours. For susceptibility testing, the reference method is the Vitic2 system from bioMérieux. It's based on the use of a 3D cartridge here that is filled with a suspension made from one or several colonies and then incubated in this system and read, and the growth or no growth of the antibiotic, of the microbe in the presence of different antibiotics and antibiotic concentration is known within, let's say, about 10 hours, depends on the situation. So the whole process takes between 2 and 3 days. So the result comes much after the clinician had to make a decision. So the question that we're asking is, can we provide more information in the beginning? And one way to do that is to use what has been used for a century now, more than a century, which is a microscope, to have information about bugs. The thing is that when just using a classical microscope, you have this kind of images without staining or with a gram staining here. You can have more discriminant information, but the level of information that you get is not at the level of what you get here. So the idea would be, can we use the same kind of methods here, which have phenotypic methods, and provide information that is relevant to what is obtained at the end of the full process. To do so, we would need to have analytical methods and global processes that would provide information on single microbes and population of single microbes directly from the initial sample. So now, how did we tackle this challenge? So as we will see, the analytical tool that we have chosen to do this identification and susceptibility testing is Raman. Raman is known by chemists for very long. It's inelastic scattering of light that provides information about the chemical bonds of the material that is probed. This has been used by microbiologists for decades now to provide information about identification and microbial biomass, and more recently a number of groups, including ours, has shown that this methodology could be also applied to very faint amounts of microbial biomass, and namely single bacteria. We have to not only use Raman, but use the whole system approach that will allow us to put the right particles of the initial sample in front of the objective of the Raman microspectroscope. The reason why is that Raman operates with a high level of confocality. So the volume that is probed is a few cubic microns. The size of a bacterium is 1 cubic micron or 2 cubic microns. So we have to make sure, in order to have the best signal-to-noise ratio, that the bacterium, the right bacterium, is exactly in the confocal volume of the objective. And to do so, we have to make sure that it doesn't move, that we are sure that the particle that we are probing is a microbe, and that it's perfectly localized in space. To do so, what we have done is first work on sample preparation, and namely immobilization of the microbes that were present in the initial sample onto an optical convenient window. In our experiment, it's a glass cover slip of 170 microns. The second part, to do so, we have to functionalize this surface with the appropriate capture chemistry. I will come back to that a bit later. Once we have done this, we have to perform a very refined mapping of the surface, and this is mapping in X and Y position, but also a Z position, in order to be sure to be in the confocal volume. For this, we have used inline holographic imaging, and I will detail that also. And once we have done that, then we can probe the different bacteria, and we by probing those bacteria, we can provide the identification information. And then if we incubate those bacteria with an antibiotic, we can also reprobe the same bacteria or similar bacteria in order to see if their chemical composition has changed, and if this chemical change can be related to their resistance or susceptibility phenotype. So first, about sample preparation. The first thing that you have to consider is that the clinical samples may be very complex. This is a urine that you expect to meet and that you see in the classical literature. When microbiologists meet with those kind of samples, they usually meet with those samples. This is urine also. I don't talk about blood. Everyone sees how complex blood can be at a particulate level. Some other samples like swabs may be a bit simpler, but even then, rectal swabs, for example, or bloody nasal swabs may also be very complex at the particulate level. So we have to put in place a method that would select the right particles, not red blood cells, not crystals, not anything, but just bacteria on a surface, be able to wash that, be able to make this happen in a reasonable time frame, because just diffusion will not bring particle toward the surface. So we had to develop a procedure, and this is a centrifuge to accelerate sedimentation, and those are some fluidic prototypes that we have used in order to push bacteria towards the surface. We have also worked on the surface functionalization, and this one, for clinical question, has to be as generic as possible. We have shown that electrostatic capture can be used in a wide number of cases, not all cases, and can efficiently remove bacteria from those kind of complex samples. This is an image that we get of bacteria captured onto the surface of our glass surface. And each dot here is a bacterium that has been stained with a live dead fluorescence marker, a commercial one. So as you can see, bacteria stick on the surface. This is a huge concentration. This is not what we have in our experiment, but it is representative. And as you can see, it's captured onto the surface and it's alive. This is another picture that is more representative of what you can get. This one is obtained with digital inline holography. And you can see the shape and precise position. So when you can probe the different bacteria that have been captured. To do so, we have first to have a refined 3D mapping of those bacteria in order to focus the Raman microscope onto those. To do so, we have chosen to use inline holography. Inline holography, without going too much into details, is interference of coherent light with phase objects that is detected by the microscope, and the interference image can be interpreted using the very well known of light propagation in order to reconstruct in silico the different Z position. So by doing that, we can have a very precise 3D mapping, and this is an image with objects in X and Y, and the color represents the Z position. Once we have that, we can use a microscope. So what we have done with our colleagues at HORIBA, the French CEA, and at the French Biotechnology Research Institute, we have built a prototype by mixing the classical SPRi base with digital inline holographic module that allows us to have mapping and analysis of the different particles in the same system, and the same system has been automated to track each particle and catch the Raman spectrum. So the configuration that we use is this one, so a glass cover slip with our microbes captured onto, and this is used for Raman spectroscopy. So the result that we obtain using that, we'll find this kind of thing classically also in the literature. What I would like to emphasize is that starting from the raw spectra, you have to develop a very refined data processing tool in order to remove this baseline. For those who are familiar with this kind of spectra of microbiological matter, this is a CH peak, this is a region where you find the most part of the information about the organic composition of cells. This is the peak where you see the contribution of glass. You have also a contribution of water. So you have to remove all of this in order to have the information that is specifically related to bacteria. So that's what we did. And this is a kind of mean spectra that we obtained on a few bacterial cells. And as you can see, this is spectra for different species, and you can see by the eye, you can see very clear differences between spectra. And when we use those differences in a statistical manner, then we can separate population of cell from one species from population of cell from other species. So this is just an example of what we had as a result. So it's a model with 8 different species. For each species is represented by at least 8 strains of the species to have a natural variability of microbiology. And when we consider means of just a few cells, the predicted species fits very well with the real species, as you can see on this confusion matrix here. So just single bacteria or a few single bacteria can be identified using this system. When we consider now antibiotic resistance, so the protocol that we have used to probe the sensitivity or resistance to antibiotics is incubating the cells captured on their surface with or without antibiotics at different concentrations, and then probing, after a certain time, probing their Raman spectra, their individual Raman spectra. The fact that we have automated our prototype makes that we were able to study this effect on very large cell population. We have done that on tens of thousands of cells. And this is a typical spectra that we obtain with antibiotics or without antibiotics. And when we compare those spectra, we see this kind of difference, which is kind of a signature of a Raman effect or antibiotic effect on Raman spectra that we've done. And when we use this signature to analyze the population of cells, what we see is that we see two effects. Either cells have a spectrum that has been modified, or they have a normal spectrum, no antibiotic spectrum. And what we see is that there is a dose effect. When you increase the concentration of the antibiotic, the proportion of the population that shows the antibiotic Raman effect increases until 90 to 100%. This is also what is represented here for a sensitive strain, so a strain that is supposed to die in the presence of antibiotics. Without antibiotics, all spectra have do not show this typical signature. When you increase the concentration of antibiotics, the population transitioned from these states toward the antibiotic effect state. And what is remarkable is that the transition occurs at a concentration that is specifically the non-minimal inhibitory concentration of the antibiotic, so the MIC, that is used, that is an information that is used by microbiologists or clinicians. And this is for the sensitive strain, while for the resistant strains, whatever the concentration of the antibiotic that is used, the cell won't die. And what you see is that the cell keeps the same initial spectrum that is similar to the no antibiotic condition. So our conclusion, our take-home messages. So first, I would like to make very clear that just Raman microspectroscopy brings information, but has to be coupled to a number of other system bricks in order to provide relevant information. So I'm talking about sample preparation, talking about mapping, I'm talking about data treatment and analysis. The second thing is that this kind of tools that is able to provide information about individuals and in large populations is something quite new and may bring new knowledge about the dynamics and about the heterogeneity of cell population that were considered up to now to be clonal and that are genetically almost clonal, but not clonal at the physiology level. And last but not least, this tool that we have developed has been developed for microbiology application, but microbes are semi-transparent, very small object imaged in water, so that's water in water, and of course other semi-transparent objects, and especially if they are not in water but in other solvents with higher optical index difference, may also be analyzable. So that's it. Before I close this, I would like to thank my colleagues from the French CEA and BioSter, and of course my colleagues at HORIBA, with whom this work has been done in the frame of projects that have been largely funded by the French government. And that's it. Thank you.